So, brief overview about our institution and our focus and our role in the Swiss research landscape. So we are not a university or a research institute, we are a pure parliament. So we are a part of the Federal Office of Economic Affairs, Education and Research, and connected to the Federal Office of Agriculture, and doing research in the ecosystem of all what is related to the food stuff. So what we see as our main drivers is in the moment driven by upcoming climate change, loss of biodiversity, but in the same sense, bringing food security and food safety for 10 million people in Switzerland soon and 10 billion people globally. Out of this part, we have a 4-year strategic program where we coordinate our research work. And so in the moment, the program is running from 2022 to 2025. So we are in the middle of this research program, and we are focusing to 6, I think I need to switch off this here. No, it's better. In these 6 focus areas, my research division is mainly acting in this area here, sustainable and healthy foods. But we have also, we define us as systems research, covering all needs from primary production on farm till functional nutrition, biology, how we as humans consume and get value out of our food. So this is driven by agriculture in changing climate, protecting the natural resources on one hand, getting most efficiency out of it, but also protecting those natural resources for the following generations. For all this production system, we are following agroecological definitions. We have also a research group looking for cost-efficient and species-appropriate animal husbandry. And in the other part, we need to look also set on the economical part, Swiss farming and Swiss food manufacturing, to stay competitive also in terms of economic sense. Just to give you a brief overview about the size of our institution, we are about 1,100 people. Based on the fact that we are no universities, also as PhD candidates we have in our institution are all based on partnerships with universities. Our output is also given in this balance that we want to create new knowledge, and the key priority of Agroscope is to transfer this knowledge as quick as possible to the practitioners, either farmers or with a special focus also to SMEs, food producing, food processing in Switzerland. You see this also, we have about, as a scientist, is key for scientific publications in the upper right, so with about 600 publications a year. But our main focus is really on this knowledge transfer. So we have also 800 practice-oriented publications and more than 1,200 presentations and posters to transfer this knowledge. What you see also is, even if we don't have a obligation to teach, because we are a pure applied research institute, but with all these collaborations we have with universities, our scientists also are embedded in a lot of teaching in Swiss universities or in universities abroad. Also based that we are a Swiss part of the Swiss government, it's key for us, research is not going in a national silo. So if you see here our research network and our actual collaborations, you see there is a focus to Europe, but we have also a lot of collaborations outside Europe with all over. So this is real driven by our research interest, where we find the best partnerships to move on with this research. In my division, the key part is this sustainable and healthy food. So we have four main areas. One is researching the microbiome in a definition from soil to gut. So we want to have the overview about the function of microbes in the soil, in the food processing, like fermented foods, but also in terms of how we need those microbes, like in our gut microbiome. On the second side, we are active in research in lowering the microbial risks and antibiotic resistance based on food. And we are promoting quality and product innovation for food as well as for feed. And we are looking, as mentioned, we see the human body itself as a biological system and doing their functional nutrition biology. Now I would like to give you some examples. So if we look to the global biomass, and this is in the moment, like in this actual analysis, we see about roughly 70% are plants. But if we look also to the microorganisms like archaea, viruses, bacteria, protists, fungi, this is huge. And interestingly, on the plants, we know pretty good. So the uncertainty is only 20%. If we look to the uncertainty in these estimates of the, and I've just taken out of the publications, the bacteria, in the moment, we even don't know the species and not the average. So we see here the uncertainty as a factor 10. So this means it could also be the majority of the global biomass could also be microorganisms, because in the moment we just don't know. And if we look to this short part of the animals, which are less than 1%, I have here just a spread out to show we as humans is just of the human biomass almost neglectable. And I think so statistic is good to have in mind when we see our following research ongoing. So with our nutrition, we focus more or less to livestock and humans, where we want to optimize feed and food, but also the health factor of this impact. But if we have the climate change in mind, we need to keep also this picture here very, very strong in mind with our activities and with our research questions. So within our institution, we have a real nice culture collection of about 80,000 isolates with a very strong focus on lactobacteria, which is used for fermentation for the famous Swiss cheese as well. But we see more and more that just having this culture collection, we need to have more information about such collections. And these are all these bioinformatic pathways which we have today. And you see the small blue, that's a part of our culture collection, and we have about 1,000 now, metagenome analyzed, sequenced. And this is the way how we need to do this microbial research, which is converting from traditional biochemistry into a more science-based discipline, which is driven by molecular biology. And seeing this, we can got such nice results. So like seeing that research out of this was the Science cover story, the most famous journal on this planet, showing what could be done with bioinformatics, and seeing how quick microbiomes are being tamed or modified. And we come back to this later when we see how we can easily change microorganisms by rapid evolution. But showing this next generation sequence analysis of our genetic resources, for us it's key to have standardized workflows, that if we partner with others, so all these strain collections are very emotional collections, and the ownership is pretty defined. But we need to find like a virtual layer, a data layer, where we can combine different culture collections. That's the reason why we have started with standard sequencing procedures, but also with the complement with the data processing subsequent, that we work on standardized workflows. And we have, this is now like open source available through GitHub, that everyone who would like to sequence a culture collection or just some strains can use this platform. And at the end, the researchers can commonly share this data platform if they're in the searching for phenotypic, genotypic research, and have open access to this data. And when you find an interesting strain out of such a collection, then you need still to have a material transfer agreement, or you need to go in contact with this. But just to have the information that there are interesting strains around makes your research work much, much easier. And so in these available functions, we have all these annotations, we have filters that you can work with this data. You can search for annotations of the phenotypic description. There are pathway maps that you can already look to genetic islands where certain functions are localized. So this is one part, because those culture collections or this processes behind to get more information about the strains is key for the subsequent work. And based on this, we look also for how can we leverage this knowledge, or how can we transfer those bacteria into the food sector. So we have actual in work a new method for the conservation of bacteria. So today you most use lyophilization for bacteria. Lyophilization is not as easy like for the microalgae, and microalgae is getting a second highly interested area of research in my department. And we have found this new method for conservation of microorganisms suitable for bacteria and microalgae as well. It's highly energy efficient. It doesn't need a cold chain, has a long shelf life, and very, very convenient in handling. And the best part, it could be used for neat materials, so just for the bacteria with a given CFU value, but it could also be used to prepare functional surfaces. So we can like coat fibers, as you see on these pictures on the right side. These are fibers which are covered with a defined small consortium of bacteria. I think this is a very nice platform for bringing applications in food, or also in feed industries, or in substrate-based plant production, to bring microorganisms into those production systems in a very convenient and easy way. Out of our culture collection, we have also a targeted research to find protecting cultures. So the goal here is to suppress the pathogens in food through feed competing with good bacteria, or which have not an impact onto the health status. You see just some examples of Clostridia in cheese, where you see so these are both all pictures are contaminated cheeses, and you see this huge mistakes in terms of evolving gases coming from Clostridia. And then you have in the middle picture a low dosage of this protecting cultures, and on the right pictures a high dosage. And you see already with a low dosage you can reduce this impact, and with a high doses you can completely suppress the negative impact of Clostridia in cheese. So we are now working with protecting cultures against Listeria and against E. coli, which are, I think, in dairy processing, the most harming food pathogens on the microbial basis, and being positive also having those cultures in this new conservation method, which is then very convenient to use them in food industry during like food fermentation, to add this, or with these functional surfaces I showed before, that we can bring this as functional surface to the inner part of food packaging, where we then can avoid the food pathogens to grow within the or in the food facing part of the packaging. Coming to nutrition, which is also part. So on a high level, I think our know-how, which is generated with our research organization, has two main priorities. One thing is to promote economy in Switzerland in terms for farmers or for food processing or food manufacturing. And the other part is to ensure with our knowledge a healthy and sustainable diet for the Swiss population. If we look to the left-hand picture, this pyramid, this is a recommendation how our society should feed herself. On the right picture, you see how the society behaves actually. So it's not a pyramid, it's more a pagoda, which is not good for our health and which is not good for the environment. So you see we have too much high protein, we have too much of snacks, alcohol, sweets, too much animal fats, too less vegetable oils, too less fresh fruits and vegetables. So this is just the actual situation, how Swiss society consuming food or defining their individual diet. If we then look to the negative consequences for health, and this is more or less globally, so the Lancet study, that dietary risks, and there is not a difference. The left-hand picture are the males and the right-hand pictures are the women. So there is not there are some differences, but the dietary risks is the highest risk factor for diseases. And this shows us that this type of diet, as we are using now in Switzerland, is not healthy for us. On the other hand, we see also, and we did this calculations, these models. So the left-hand side is how we behave today. If we would move back to the pyramid, which is shown in the middle, we would reduce more than 50% of the carbon footprint of our individual nutrition. So this shows like the win-win situation we have with changing our dietary behavior in the society, and the society needs to be trained. We need to transfer knowledge to the society for this change in our dietary behavior. And I'm also convinced that there needs perhaps some modification in the legal framework, but the main motivation should come from each individual, because it's really a win-win situation, how we should change this behavior. Again, because we are working with microorganisms. So looking to the microbial diversity, and this shows just what diversity is needed. And this is just our into the gastro part of our body. It's not what is on our skin and what we have other, but looking the biodiversity and the red dots here on this side is showing out of this genetic wheel where also different microorganisms in our gut microbiome are originated. And this diversity shows to us that it's huge. So we as organisms, the human, is much closer to any food like a tomato or on a genetic basis as those bacteria sitting side to side in our gut. And this shows us also the chances and the risks of a dietary, how we can influence this microbiome. So having here a good nutrition with a lot of dietary fibers, with a lot of phytochemicals, with all these antioxidants, with prebiotics. On this side, the diet as we have today, with a low amount of dietary fibers, with a lot of processed food, high amount of red meat, out of this, a huge range of obese people. Having this, we see the host benefits to our gut microbiome. We got a lot of, in this blue part, as positive part, we have a lot of micronutrients, gut immunity. On this negative side, we have all this supporting these effects. And with this diet, we are reducing those risks. So we have no benefit here, and we are supporting bad effect. And this shows us, if we compare this with macroscopic view, if you see this here as a picture of a good gut microbiome with a huge, and these are those alpine meadows where Switzerland is famous for, which are really very rich, very diverse. This is also translated because we can't see our gut microbiome, and I want just to take this as a comparison. This is how our gut microbiome should look like. With our strong, this Western diet, as we do, the gut microbiome is coming into something like this. And when we then try to reconfigure this with a change of dietary, it's not easy to go from this side back to this side. The probability that we go to this side is much higher, and not having this diverse, just very limited. And on the right side, we see how this happens. So if we have this westernized diet, as we have shown in this pagode, we enrich this red, it's a very small number, and we deplete the green ones, which we should have. So we are going more into such a pictures and going back in like such a picture. I hope this comparison helps you how we try to do our research for how can we drive and promote a good gut microbiome by the impact of nutrition. And this is also for us one of our actual research projects, which is named Fair Food, where we want to promote consumes of fermented foods. So we have shown that with selection of beneficial bacteria and fermented foods with a potential for this nutritional and health benefits, that we can drive the quality of fermented foods, which is very easy for the gut microbiome to handle with those nutrients, and then wanting to transform this into a public health part, to bring this fermented foods into the Swiss diet, with also based on nutrigenomical research to show the effect of this change. Today, nutritional science is very much based on experience, like medicine as well. And what we want to do here with biomarkers is to show in nutrition research with methods, as we know from clinical diagnostics, which is really evidence-based science. So if we find those biomarkers for a good and positive nutrition, and find biomarkers for a not so good nutrition, helps us a lot in defining the new dietary recommendations, and then bringing this to public health. What we also do is just looking into like all these alternative novel foods which are approaching the market, like with a lot of alternatives for animal-based food, which are now designed with plant-based foods. So this picture shows just what we got in the shop as alternatives for cow milk. And on the right hand, you see a scheme of a process, how we get out of lupines, so a protein isolate, which is then used to formulate such a protein drink. And what we see is that in this comparison, only the soy milk is roughly comparable this does not mean that these other products are not good food materials, but you need just to take in consideration why you take individual food into your diet. And if you compare this, so the protein content, so cow milk and soy-based alternative is pretty comparable on the protein content and also in terms of the minerals and micronutrients. But if you look to this other, like oat milk, or which is the protein content is on less than 15% of what cow milk has. And this is also when you then look to the life cycle assessment of those products, the life cycle assessment is normally done on a weight base. And I think the life cycle assessment of food materials should be done on a nutrient base. Because why we take those food into our diet? Because we need those nutrients, we need proteins, we need calcium and other micronutrients. And when we want to compare those products, on one thing, we need to compare them on taste and on the physical behavior, like the milk products. Are they ready to prepare a foam or something like this? But for me, the key priority is what is the nutrient density of those products? Because only with this knowledge, I can integrate them wisely into my individual diet. Another part I mentioned is how to find out this evidence-based. So how do we find biomarkers which represent a good nutrition? This is in the moment mainly done with methods, as we know, from clinical diagnostics, where we take urine or blood as biomatrix, but these are special. The blood is an invasive method. We are now in a research consortium where we explore the exhalome. So what we press out, to use this exhalome as biomatrix to find metabolites. And you see, this is a very wide consortium of research institutions, where we are part of a lot of research institutions like EMPA or ETH or Paul Scherrer Institute, but also most of the university hospitals. And we are here focusing with our activities into the metabolomics part. So also a little bit in the microbiome, nutrition science, but we want to see, or our goal is to find in the breast nutrients, metabolites which represent a good nutrition or a not so good nutrition. So this is just shown the high resolution mass spec platform to do this. There's a breast is coming from here, the CC source here, and then we find those metabolites. And we are not only interested in the humans, we also apply this technology now to our animals. To show special here a picture for ruminants, where we show the effect of different diets in the exhalome sampling and looking for metabolites for like exhaled volatile fatty acids or methane emissions, which are in the ongoing research for the ruminants quite high priority. The last part in my presentation, I would like focus to microalgae, which is also, in the moment I've shown the role of microorganisms for food fermentation or in terms of food safety, like that. But now I would like to go into microalgae, where we see the microorganisms themselves as very effective bioreactors for real high-value nutrients like proteins or omega-3 fatty acids or lipids. And the main interesting part of those microalgae is that you can integrate the production also bioreactors into buildings, so without any use of arable land. And this is our goal, that we can, like in the livestock part, that the farmer can produce the high protein feed materials themselves, so like a vertical integration also in terms of the value chain. And on the other part, that you really do not use arable land to produce feed material. So we see also that arable land should be used to produce directly for human nutrition and not for feed materials. So just going into the area of microalgae, it's such a huge biodiversity, which is in the moment perhaps to a quarter explored. For the rest, especially in the aquatic systems, in the marine systems, we just do not know in total what are around as species. And just looking also to the microscopic picture, which I think is a huge artwork of nature, seeing the biodiversity of microalgae. Then if you compare them with glasses of a scientist, if you produce soybeans on a field, the best breeds, and we do also plant breeding and Agroscope with a focus to adopting soys to Swiss climate, but with the task to use soy directly for human nutrition. On 1 hectare, so on 10,000 square meters, with the best soy breed we have today, we can produce protein to feed the protein demand of 40 people. If we take 1,000 square meters and do a photoautotrophic cultivation of microalgae, which means we do not need arable land. This could be roof surface or any area. With this, we can produce the annual demand of 520 people. It would be even better to plant sugar beet and to convert this with microalgae into protein. So this just shows the efficiency of this single cell bioreactors of microalgae or other microorganisms like yeasts or fungi as well. Also looking to the scheme of the life cycle assessment of having centralized production of feed materials, where you need fertilizers, plant watering, electricity for transportation and for processing, labor you need for, and then bringing this to the livestock part, comparing this to a photobioreactors which are integrated on farm to the existing buildings. We haven't quantified this yet. That's part of the ongoing projects. But just seeing the scheme of the LCA shows you that there's a huge advantage to be better with those on-farm landless production of nutrients for animals as well as for humans. This shows where we are today in this. So we have scaled up from the flask to 200-liter tubular photobioreactor. So we have already shown a scale up of 2000. But when we want to bring it to the buildings, and if we calculate, so for an average Swiss farm, we need like a photobioreactor volume of 50 to 70 cubic meters. There is still work to do, which we are happy to do. The other part of this nutrients by microalgae, we all know that salmon is very rich in omega-3 fatty acids. But these omega-3 fatty acids are not produced from the salmon. It's produced from the microalgae, which is feed of the salmon when he is in natural condition. When we aqua farm the salmon and feed it with soy powder, you see here how the content of omega-3 fatty acid decreases in aqua farmed salmon. Now it's increasing again because also in aqua farming, processed algae are used to feed the salmon again. But the cool part is seeing this result. So if we feed pork or beef with microalgae, we get the same amount of omega-3 fatty acids in beef or in pork meat as we have in salmon. And so we can add not only the advantage of not using arable land, but also having a nutritional benefit to animal-based food material. So that's how we want to integrate this in our research. So this is in the moment under construction for our new research campus. That's animation of artists who I know personally, who showed me some visualization, how such bioreactors could be integrated into buildings, that we don't have this competition feed food, which is actually ongoing. And this was rough, my overview to the Swiss, how Agroscope engages this in the research for food, feed, and nutrition. And now I'm happy to discuss